For those that don't know me, my name is Niv Dagan. I'm the Executive Director of Peak Asset Management. We're a boutique investment management corporate advisory business, Melbourne-based, love biotechs. And thank you very much for everyone for attending, and Rudy and Verity and the team. I've got some great speakers tonight, two companies of which actually we're both shareholders of. Firstly, I want to welcome Tim Oldham, the CEO and Managing Director of AdAlta. Tim, and I've just written a few notes to introduce Tim. 20 years experience in small and mid-tier life science companies, spanning business, product development, commercialisation across Europe, US and Asia. Prior to his appointment as CEO in 2019, he was executive leader of Tigen Ventures, an advisory business focused on growing life science companies, and CEO of Cell Therapies Pty Ltd, a contract manufacturer of cell and gene therapies. Tim currently serving as a non-direct executive director as well of ACRAX and director of Bio Melbourne Network. For those who don't know about AdAlta, AdAlta is developing a proprietary iBody platform to solve limitations of antibody drugs and target challenging medical conditions. Some of the key assets include a phase 2 ready, AD214 for the degenerative fibrotic disease, and 4 partner discovery stage assets in immuno-oncology and CAR-T cell therapy. So with that introduction, I might bring Tim over here to tell us a little bit about the company. And Tim's on the road as well, roadshowing the company over the next few weeks. And Peak, which is us, we're involved in placing the shortfall of a rights issue. So for those interested, come see me afterwards. But welcome, Tim.
Thank you, Niv, and thanks everyone for joining us on this wet and dreary Melbourne afternoon. With a little bit of luck, we've got a way of getting the presentation up here. Okay, so as Niv mentioned, AdAlta is all about developing next generation protein and cell therapeutics with the objective of solving debilitating diseases where traditional antibody drugs haven't been successful. In essence, we are trying to go where traditional antibody drugs cannot. Antibodies transformed pharmaceutical practice about 30 years ago when they first hit the market, giving us the ability to very selectively and specifically hit drug targets that small molecules couldn't, but they can't do everything. And our iBody platform is specifically designed to do the things that antibodies can't. We succeed by producing a high-value pipeline of next-generation protein and cell therapy products. We have 2 parts to our business. Our discovery business, which is depicted on the left-hand side of this slide, is all about our iBody platform and is about generating iBodies or binders that hit these difficult-to-drug targets. These become our saleable inventory, if you like, the stock we have on our shelf that we can then continue to develop into products ourselves, or we can partner with other companies to do that. And we have programs in fibrosis and inflammation today, as well as programs in immuno-oncology. So this slide in one page really summarises the impact we are trying to have on human healthcare today. We're trying to solve the challenges of fibrosis. These are degenerative, progressive, and usually fatal diseases of multiple organs. In common with our colleagues and partners, as you'll hear more about shortly at Carina, we're trying to work to take the promise of CAR-T cell therapy to provide new hope for patients suffering from solid tumour cancers, building on the success we've already seen in blood cancer patients. We're working with our colleagues at GE Healthcare to help better target new immuno-oncology drugs. I don't plan to talk very much about this asset today, but essentially what we're trying to do is develop PET imaging agents so we can tell whether you're one of the 1 in 5 people who is responding to these new checkpoint inhibitor drugs within weeks of you starting therapy. More importantly, we can tell you if you're one of the 4 in 5 who's not, and we can tell you that before your tumours progress. And finally, our platform, which underpins everything that we do, is itself a partnerable or saleable asset, because companies come to us and say, can you do X? And if we think we can, that leads to additional collaborations. All right, as Niv has mentioned, we have just closed a rights offer. We announced the results of that this morning. We're targeting to raise 3.15 million, and I was very, very happy with the 1.28 million we've raised from our existing shareholders. As announced this morning, the remaining shortfall of 1.87 million is already covered, but there are opportunities for new shareholders and new investors to participate. As Niv has mentioned, please contact the Peak Asset Management folks who are in the room today, should that be of any interest by the time we get to the end of this discussion. So let me talk about AD214. This is our most advanced asset being developed for fibrotic diseases. So I'd ask you to think about what happens when you cut yourself. Right, your body repairs. It repairs by creating scar tissue to pull the sides of the wound together. But that leads to your skin puckering and getting stiffer. Right? Now that's a normal wound healing response that we're all used to and familiar with, and those of us who are all getting a little bit older, that takes a bit longer than when we're as young as some of the younger people in the room. Now imagine if that happens in your lungs. Your lungs expand and contract every second of your life. And if that starts to stiffen up, then your lung capacity, your breathing capacity is going to be severely diminished. Now this happens for about 500,000 people around the world today. We don't know what causes a disease called Idiopathic Pulmonary Fibrosis, basically scarring of the lungs. But we do know that it is degenerative, progressive, and ultimately fatal, with half of patients diagnosed with IPF dying within 4 years of initial diagnosis. We, globally, spend about $4.3 billion a year on drugs to treat this condition, and those drugs don't work. They slow progression modestly, and they have some very serious side effects that prevent most patients tolerating them for more than about 6 months to a year. Not a great outcome with a 4-year life progression. So there's a desperate need for new drugs to treat this condition. Beyond lung fibrosis, we're also looking at fibrosis of almost every organ system. Lungs, kidneys, eyes, heart. We know about long COVID. There's a fibrotic component in long COVID. I'm sure you've all heard about the recent uptick in silicosis cases. Silicosis is another scarring of the lung disease. Now, we don't know that AD214 is going to work in all of those applications, but we think that fibrosis is a condition that applies and it's got similar mechanisms right across all those organ systems. So, what have we done to date? We've actually developed a first-in-class, and this means it's the first molecule to target this particular disease pathway, which provides a competitive advantage. We've developed a first-in-class molecule through the end of phase 1 clinical trials. We've validated the mode of action. We have preclinical efficacy data in a wide range of animal models. We've got a manufacturing process that's already been established. We've successfully navigated phase 1 clinical trials to demonstrate its safety. And then we're working with a strong intellectual property position with patent protection out to 2036. So we believe this is a compelling opportunity for larger biopharmaceutical companies seeking to add established IPF assets into their pipeline. So our next steps are to take this molecule into phase 2. We plan to do that through partnerships, and our partnership discussions are accelerating. We have a number of companies under CDA evaluating this asset at the moment and asking increasingly sophisticated questions, which is always encouraging when you get to that stage because it means they're taking serious interest. As Niv had mentioned, we're on the road for the next 2.5 weeks talking to potential pharmaceutical partners at Bio International in Boston, ASCO in Chicago, and visiting several other partners in the UK on the way home. In parallel with that, we're planning and preparing for the phase 2 intravenous study of AD214. We've secured our manufacturing slots. We've secured the toxicology study slots that we need to do to run that study. And the money that we're raising at the moment is enabling us to take AD214 back to the clinic earlier than we would have previously anticipated in a phase 1 extension study that will actually help our partners with additional data to inform the phase 2 study design and ultimately shorten the phase 2 study for them. So we think that adds significant value to the asset as well as generating that new data to enhance our partnering efforts. This study is summarised on this page. Essentially, it's evaluating safety at a higher dose than we've previously looked at. That will result in a shorter and lower cost phase 2 study. It will reinforce the safety trends that we've seen so far and enhance the partnering process with additional data and maintenance of our ongoing product development momentum. If you like, we're keeping the inventory, the saleable inventory on our shelf fresh. And why does all this matter? Because big pharma are paying significant amounts of money for assets in this stage of development. This slide summarises recent licensing transactions by big pharmaceutical companies for IPF assets ranging from preclinical through to phase 2. AdAlta's AD214 sits somewhere in the middle of this pipeline, and you're looking at the upfront payments that have been made to licensors along the way, and you can see that there are some very substantial figures there, particularly when you think that our current market capitalisation today is less than $10 million. So the upside potential of us being able to secure a transaction for this asset are significant. But AD214 isn't the only asset that we have in our portfolio. We are co-developing with our colleagues at Carina new iBody-enabled CAR-T cell therapies. Now, the opportunity here builds off the transformational outcomes that have been achieved for patients with blood cancers with CAR-T cell therapies. Deborah, I'm sure, is going to provide a much more detailed overview of what CAR-T cell therapies are. But essentially what we're able to do now is reprogram a patient's own immune system to be able to see the cancer that's been hiding from it. CAR-T cell therapies are living drugs with the potential for a single dose being able to cure a patient from cancer. The outcomes for paediatric cancer patients in particular have been stunning. The little graphic on the top right-hand side of this slide is a nod to Emily Whitehouse, who was the first child treated with CAR-T cell therapies back in 2012. She's just celebrated her 18th birthday and 11 years now cancer-free. One dose of a living drug. So these drugs were first approved by the FDA in 2017. They're already generating over $2.5 billion in sales a year. The market is forecast to reach $20 billion by 2028. But the problem we've got is that 90% of cancers are not blood cancers. They're solid tumours, and CAR-T cells have yet to be successful in terms of marketed products in this field. Yet half the revenue of that 20 billion sales is going to come from treating solid tumours. And that's where AdAlta's iBodies and Carina's technology come together to create better CAR-T cells with potentially better precision, performance and persistence to deliver outcomes for these solid tumour patients. So the advantage of the iBodies in this picture are their small size. iBodies are tiny. They're a fraction of the size of a monoclonal antibody, less than 10%. They're half the size of the typical binders that are used to create a CAR-T cell. And that means we've got twice the capacity in our, what we call our gene cassette, twice potentially twice the functionality of the CAR-T cell than you could achieve with traditional technology. When you combine that with Carina's capability to provide long-lasting, highly potent CAR cells, we can provide the ability to target those CAR cells with unique tumour antigen targets, and also with bispecifics or multispecifics that enable us to target multiple tumour antigens at the same time. That gives us better targeting, better persistence, and potentially the idea that the T cells could also secrete their own molecules to overcome the immune suppression of the cancer. So we think this is a really powerful combination. We also think it's possibly not limited to the work we're doing with Carina. We've committed to up to 5 targets with Carina, and we're very excited to be working on 3 already, with the first of those going into animal studies as we speak. We had significant industry interest from other players in the field around the world. And these assets, again, are valuable when we get to the end of preclinical proof of concept. So that's very early without even going into a patient. The deals shown on this page were all done with assets that have not yet been in the clinic. So once again, significant upside value from transactions here. So I've talked about the 2 major parts of our pipeline today. AD214, our clinical stage asset, the 3 programs we're already working on with Carina with the potential to expand that to 5. Our collaboration with GE Healthcare for PET imaging I mentioned right up front. That gives us our 5 programs that we're actively working on. At the bottom of this slide you can also see that we have a couple of other assets that we're not actively developing at the moment, and historically we've been able to demonstrate the ability to hit over 25 different drug targets with our iBody platform. So this gives us significant additional potential and drives us down a partnering program that has 3 main pillars to it. And I'll touch on that as we go through our investment proposition. So our iBody platform is the core engine room of our company that allows us to hit drug targets and in drug formats where traditional antibodies have been unsuccessful. We have our lead asset already in the clinic and being prepared for phase 2. And our strategy for that asset is to now realise a return on the investment we've made in that asset as we seek partners to help us take it through into phase 2. We have 4 immuno-oncology programs underway, 3 of them with Carina Biotech already, and our strategy here is to progress those programs that we've already started and to expand the number of programs and the number of collaborations that we're working on. We've demonstrated through those 4 programs and our partnering that we have the capability to both develop products and to partner those commercially. And as we've seen, I think the blue sky potential from transactions around our existing assets is significant. And on top of that, we're also working on a second pillar of transactions, which is expanding our Carina and GE-like collaborations, and a third pillar of transactions, which are around looking at complementary technologies and assets that will be enabled by our iBodies down the track that we may have the opportunity to bring in-house. So significant blue sky opportunity, we think, through inorganic growth opportunities. We've got a fairly steady news flow coming through over the next 6 months, even without the transactions, and we look forward to reporting by the end of the year on the results of our phase 1 extension study that the current capital raise is financing. As I mentioned, we do have the current raise covered, but there are opportunities for new investors to continue to participate in the shortfall placement. And with that, I'll stop and see if there are any other questions. Thanks very much for your attention.
Sorry, can you just explain how the iBody platform actually works?
So iBodies are what are called a single domain antibody. They mimic the structure and features of antibodies found in the shark. We've engineered that onto a human protein that has exactly the same structural configuration. And we have a library of 10 billion variants of these iBodies, all of which will bind potentially to different targets. And so it's a fully synthetic, fully human, single domain antibody platform in technical terms. Basically think of it as a soup of lots and lots and lots of very, very small antibody-like molecules. And then we screen that against targets of interest to find iBodies that bind to that target, and where we need to induce certain pharmacological effects, we have added screening to enable that to happen. So we own that technology, we own the IP, and we're the only ones, we're the first to produce a human version of that single domain antibody framework.
Thank you. And the additional, the additional genes that can go into that iBody, is that all, like, in terms of the application of how you administer that, is that all in one go, or is it then you do the iBody first and then once it's set up then you can administer the additional bits? So
there's a couple of different ways we can use the iBody technology. We can use it directly as a therapeutic, in which case it can be directly injected, or we can do certain things to extend its half-life, which is what we've done with 8214. So that's a protein molecule. It's used directly as a therapeutic, much like a traditional monoclonal antibody. When it comes to technology like our CAR-T cell therapies, what we're trying to do is engineer the T cell, so that requires genetically modifying it. The iBody is part of the gene cassette that is introduced, or the gene sequence that's introduced to the T cell to express that CAR receptor that allows it to go find the cancer. So the iBody is part of that gene cassette. Everything else in that cassette that has the linker into the T cell and the signalling sequences within the T cell itself are all part of the same gene cassette. But you can only put so much genetic information into that cassette. So the smaller the iBody component is, the more bits you can put in to create those multifunctional CAR-T cells.
Awesome, thank you.
Tim, 2 questions. One is, what would you say sort of your unfair competitive advantage on a global scale And the second one, obviously there's a lot of upfront with some of these partnership opportunities, but what would you say, what does a partnership look like in the eyes of AdAlta?
Well, there's a couple of very big questions there. So what's our advantage? So we're the first fully human single domain antibody-like system. The advantage of that is that the other systems are based on shark or camelid, camels, llamas, alpaca species. They potentially have a greater risk of an immune reaction by a patient to the drug. So that's our advantage over those other technologies. Our advantage over the monoclonal antibody field in general is that the structure and features of these iBodies enable us to hit many different classes of receptor that traditional antibodies can't. So monoclonal antibodies are really good at hitting soluble proteins that are circulating in the blood, or hitting targets that are expressed on the surface and sticking out of a cell. But there are many, many classes of drug target that don't look like that. For example, we target with 8214 a receptor called that's part of a family called G protein coupled receptors. They're a bit like email inboxes for your cell. They tell the cell how to respond to the external environment. But what that means is that the receptor is in the cell membrane, and the place you need to bind is buried in that cell membrane, and our iBodies have much longer binding loops that can get into those cells. So they're the sort of real features, the modularity and the ability to hit these different targets. What does a partnership look like for us? There are probably 3 different versions of a partnership that we're interested in right now. Version number 1 relates to 8214, where we'll be looking at a larger pharma or biopharmaceutical company to essentially license or co-develop the asset with us. That means funding the development and the cost of the phase 2 clinical trial. And they would then carry it through into phase 3 development and commercialisation. That deal would usually attract an upfront payment to us and then further milestones down the track as we get through clinical and commercialisation and royalty payments on sales. The other types of collaborations we're looking at would be co-development collaborations from the get-go, such as we have with GE Healthcare and Carina, where we either jointly fund the discovery effort to produce an asset that becomes saleable, or in the GE case, they fully funded our discovery processes. And so that's where we're looking at the moment with other CAR-T collaborations, is for folks who can work with us on that sort of basis. All right, thank you very much.
Well, thanks Tim for that excellent introduction. So let me tell you about our CAR-T technology and some of the history of Carina Biotech. So we have a history that goes back now over 10 years in terms of CAR-T generation and manufacture. We're a spin-out from CRC, the CRC for cell therapy manufacturing, that brought together researchers across Australia, all with an interest in cell therapy manufacturing. So a very deep history underpins our CAR-T technology. The platforms that we've developed over that period of time include a manufacturing platform, which we believe is the world's leading platform for autologous CAR-T generation. In fact, we are now competitive with any of the so-called off-the-shelf technologies. So a 9-day manufacturing process amongst a field where 15 to 30 days is common practice. Humira, for example, is manufactured in 17 days. This not only reduces the cost of manufacture but also benefits the patients. You get the cells to them quicker, and these cells are in better shape with higher transduction levels. Our lead program, which I'm excited to say will be kicking off with first patient enrolment fairly soon, currently evaluating a couple of patients for entry into that trial, will target a cancer stem cell antigen called LGR5, and I'll show you some of the exciting data that we've generated preclinically that has supported our successful IND filing. In fact, we were surprised to get that IND filing ahead of our 30-day review period by the FDA. So surprised that we hadn't prepared a press release for it. And being a privately held company, that didn't matter. That really didn't worry us too much. We were able to take our time. So successful IND Phase 1/2 trial that was cleared through that IND with the FDA about to commence with first patient enrolment, and a very strong shareholder base that includes 10 Mile Ventures and the impact fund of the Minderoo Foundation. We've recently been in fundraising mode, which has been very successful for us, and we are about to close on a $10 million component of that raise, which was strongly supported by our shareholders as well as some new investors. We have a very strong patent portfolio, as you would expect, and so without further ado, let me walk you through the Carina Biotech story. Although Tim's told you a little bit about CAR-Ts, and some of my slides might look a bit familiar, I will cover the territory again. So CAR-T therapy is a very personalised therapy where immune cells, white blood cells, are taken from a patient with cancer. These cells are then engineered to generate antigen binding domains. They are constructed in a way that will help them track to the solid cancers and then reinfused into the patient. So a very personalised therapy that empowers cancer patients to fight their own tumours. There are 6 FDA approved products, and I'll show you some of the revenues that the leaders in the field have generated. But these are all for blood cancers. And so the challenge is for CAR-T therapies in solid cancers, which of course comprise about 90% of all cancers. And of course the field was set alight with the acquisitions of Kite Pharma and Juno Therapeutics some time ago by Gilead and Celgene for quite large sums of money, and it's been encouraging to see as we've moved into more difficult times in the market, and again very pleased to be a private company at this point. We've seen still some very strong deal making in the space. And by 2030, it's projected by analysts that some 50% of the revenues from CAR-T cell therapies will be derived from solid tumours. So currently there are no therapies that are available for colorectal cancer, which is our indication for our LGR5 targeted CAR-T cell. But there's lots of evidence that even in a more mature therapeutic indication that CAR-T cell therapies can really revolutionise treatment and take over from the older therapies. And the first CAR-T therapies approved, Yescarta and Chimeric, certainly prove this point. Whilst they're pricey, if they work, they are curative from a single inoculation of the CAR-T therapy, and this of course has significant economic and other benefits for society, as well as major impact on patients. So reimbursement is supported, for example, in the US and other major markets. So we think there's a great opportunity for our CAR-T targeting LGR5 in metastatic colorectal and other solid cancers, but we recognise the challenges, and this has really been a focus of the integrated approach that Carina has taken since its inception. We've recognised that precision of targeting of cancers is very important. The target needs to be very well differentiated between the cancer condition and what's seen on normal cells. And for this reason, we've targeted cancer stem cell markers in our approach. And we have one of the most innovative pipelines of any of the cell therapy companies that are seen worldwide. So we're not just another CD19. There are so many CD19 companies out there. The second challenge is getting the CAR-T cell to see the solid tumour, getting them into the solid tumour. And we've developed a proprietary chemokine receptor platform. Our CAR-T cells contain an additional engineered element to the CAR, and all cancers, all solid tumours produce chemokines, and this sets up a chemokine gradient. So the further you are away from the solid tumour, the lower the level of chemokines. And as you move closer to the cancer, the level of chemokines are increased. So the engineering of a chemokine receptor, so something that will measure these chemokines and track towards them, is an important component of our positioning. It certainly also enhances potency of killing because it gets the CAR-T cells to the solid tumour quicker. The other element that enhances the potency of Carina's CAR-T cells, and I mentioned our background in cell therapy manufacturing and adopting all the innovative approaches from the CRC years, is our manufacturing process. I mentioned that it's shorter, but the advantage is that it's producing more potent CAR-T cells. And these are very robust CAR-T cells. Less than 0.1% of them show markers for exhaustion, which is a key feature of any release criteria from a GMP manufacturing process. And also they're capable of waves of killing. So we call them serial killers. There's the phenotype of cells that are generated in our manufacturing process. There are ones that go on the immediate attack. There are the follow-on CAR-T cells. And then there are those CAR-T cells, a very small number, that become embedded in lymphoid tissues, including spleen. And these are the CAR-T cells that we believe, and certainly our data indicates, are available should cancer recur. They are then reactivated sufficiently to fight the cancer. So hence the waves of CAR-T killing and the serial killing nature of them. So they're better than an antibody. An antibody is consumed, kills one kills one cancer cell, and then it's done. Typically, a CAR-T cell will keep killing multiple cancer cells in multiple waves. So this is a diagram of our manufacturing process. As I said, it's a 9-day process, amongst the shortest of the industry. We're certainly world leading in that approach, and the feedback that we've had from potential partners is that we, as I said, are very, very competitive. For example, Chimeric is manufactured in 17 days. Other CAR-T products manufactured by companies that are currently in phase 1 to clinical trial, we know take 28 days to be manufactured. So we are very focused on getting the best out of this manufacturing process. It's not just about the time, but it's also about the quality. And we're getting over 90% transduction in our LGR5 program. This means that over 90% of the cells that come out of our manufacturing process are actually CAR-T cells. This is not always the case. For some products it's as low as 10%. In fact, the release criteria for Chimeric in fact is 10%, and for other companies it's as low as 30%. So we're certainly industry leading in terms of the overall CAR delivery efficiency. So the shorter time, the quality of the cells, the higher level of CAR cells containing cells in our manufacturing process, the lower levels of exhaustion, and the ability to create a CAR phenotype which we monitor through our manufacturing process that enables this 3 waves of serial killing. So this is a cartoon of our manufacturing of our CAR-T cell. We make CAR-T libraries and select the best one as a clinical candidate. We engineer the antigen binding domain. We engineer the optimize the linker domain. So we don't just make one CAR-T cell and take it into the clinic. We actually select the best one, and I'll show you why we've selected CNA3103. A bit of a mouthful, but it's our candidate for the clinic. You can see also, in addition to the antigen binding domain, the optimized linker domain, there are intracellular signaling domains. There are also some intracellular aspects to our CAR-T, which are not shown in terms of signaling on this slide, but also you can see the additional chemokine receptor that we put into our CAR-T cells. So let's talk about our lead program, LGR5. It's a cancer stem cell marker. It's been researched for the last 23 years or so. It's known to be an initiator of certain types of solid tumours, including ovarian cancer, gastric cancer, as well as metastatic colorectal cancer. LGR5 expression is known to be an indicator of more aggressive forms of neuroblastoma, and there's various other independent research on LGR5, and much of it led by the current head of R&D at Roche Genentech. So they play an important role, these cancer stem cell markers, and LGR5 in particular, not only in initiation of cancer but also in progression and metastasis. So the evidence in the literature is that their depletion of LGR5 cells in colorectal cancer restricts primary cancer growth, and LGR5 depleted cancers are not able to metastasize. So that's why we're so excited about the target. We're the only company in the world that's developing an LGR5 CAR-T. There is a bivalent LGR5 antibody that's in clinical development and presented some very interesting data that was the subject of a plenary talk at ASCO last month. But the important thing about cancer stem cells and about cancer stem cells bearing LGR5 is that they're not killed by chemotherapy or radiation. So, very, very valuable addition to the approach that these agents are already undertaking. Colorectal cancer is on the rise in certain patient populations. It's now being diagnosed in people who are younger and younger, in their 20s, 30s and 40s, at a late stage. And it's in this patient population that tend to be LGR5 positive. Somewhere between 70% and 90% of these patients are LGR5 positive. And it's still the second leading cause of cancer death. It's still the third most commonly diagnosed form of cancer. So this is just showing you that differentiation that I talked about. You want a target that is not highly expressed in other parts of the body. There's a very restricted expression pattern for LGR5, but you want it to be really highly expressed in your cancer setting. So here you can see on the, I guess it's the left-hand panel, depending which way you're facing, you can see a normal colon, very, very high magnification, barely any LGR5 showing up, as indicated by the arrows. That brown stain in contrast to the brown staining that's seen in a liver biopsy, and liver is the first place that colon cancer metastasizes to for most patients. You can see very low magnification. Is that the 14-minute wave? Okay, cool. So very high levels of LGR5. So this is one of the pieces of data that makes us so excited about our LGR5 CAR. It can clear tumours very quickly from animals that are bearing, in this case, a colorectal cancer. This is a very durable response, and you'll see here in the yellow line, that's our clinical candidate, CNO3103. We tried many other LGR5 candidates. This was the one that gave the most durable response from a single administration. And then this is also the one where you can see absolutely no tumour recurrence when you re-challenge the animals with fresh cancer cells. As I said, we've got a phase 1/2 cancer trial that will start in the next week, 3 weeks, but very, very soon. And in the phase 1 component, which is a Boyne design, we'll be looking at the recommended phase 2 dose level, and we expect to be completing that component of the trial around end of quarter 2 next year. The phase 2 dose will then proceed, phase 2 trial, will then proceed seamlessly, and we expect that enrolment will be completed around the end of 2024. So should we choose to go public, obviously we're looking at the current raise as a potential IPO raise, then we'll go public with some clinical data. Of course, we're not leaving any of our doors closed around other options, whether that's licensing or acquisition of Carina Biotech. You can see a little bit more here, the detail of our clinical trial. The dose levels cover a very broad range and begin at the lowest dose range that we've seen activity in animals. This speaks to those recruitment milestones that are forecasting, obviously, until you actually get into the clinical trial and begin recruiting and know where you stand. You never really know, but this is what we think it will look like. And this is our broader pipeline, so lots of opportunities to expand the pipeline in the longer term. And I'll just leave you with this final slide that summarises Carina Biotech. Thanks for your questions.
Over here, Deb. So with the LGR5, do patients generate natural autoantibodies to that? Is that how you filter your patients?
No, they don't. They don't generally develop natural antibodies to LGR5. And there again, other companies have done LGR5 antibody, exogenous antibody therapy, and they generally no autoantibodies of note.
I'm not familiar with the mouse model. What sort of intact immune system do they have? Or
well, because we're evaluating a human cancer, these and we are adding human CAR-T cells. Of course, we're using an immunosuppressed animal, so we're not looking at any kind of internal immune response that might be generated from the animal. We're looking at purely what our CAR-T cells do in that model. And we've looked at low expressors, high expressors, medium, intermediate expressors. The data I showed was actually from an intermediate expressor, so that we've got a good understanding of the dose-response relationship in those different settings.
I was just looking at your potential endpoints for phase 1, 2A. Is that anything to do with tumour reduction or longevity?
Yeah, so
the FDA giving you guidance on where you should be?
Well, they've approved our clinical, well, given the green light to our clinical trial, so the answer is yes. So the phase 2 primary endpoint will be overall response rate, and obviously response will be measured by RECIST, and also, so that's RECIST 1.1, but also by iRECIST.
What's RECIST?
So that's the criteria. You're actually measuring using MRI the size of the tumours. And there are two types of, it's called RECIST criteria, and it's just a way of measuring the size. And there are two types. One is used for measuring immuno-oncology agents, and that's called iRECIST, and the other is used for measuring all sorts of anti-tumour products, the response to tumour products. It's been in use for decades, really.
So your competitors are in the space that not do those hurdles?
Well, in terms of competitors, in terms of addressing, there's no one doing LGR5 or actually any of our CAR-T cell targets within our pipeline. There are no competitors that have demonstrated preclinically dramatic improvement in their manufacturing process, impacting CAR-T potency, for example. And we know that really we're pretty world leading in our manufacturing process. I mean, there are a lot of CAR-T companies out there, and it's interesting. When we started looking at LGR5, we were sort of the new kids on the block in terms of taking a CAR-T product for the first time into the clinic for LGR5. And as we've progressed our program, I think it's fair to say that we've been working at a pretty rapid rate, even though we went through the IND process and got that approved. And we're now at a stage which is fairly similar to some of the other CAR-T companies that kind of started a bit earlier than us. Yeah, so we just have to keep, you know, running ahead of the pack, really. That's our aim. I think there might have been a couple of other questions. Rightio. Thank you. Thanks, everybody.
Thanks, Tim and Deborah. Two very interesting stories. A nice little uplift on our education on CAR-T as well. Two great endeavours, and that's why we love biotech. We love stories like these, and we've got a good rate of success of it for a lot of these companies in Australia. I'd like to also acknowledge FB Rice and thank FB Rice, our sponsors. And now I will invite you all to join us for some refreshments outside. Thanks everyone for attending and braving the rain to get here too, by the way. Thank you.